In this video, we're going to talk about prokaryotic translation. Before we talk about the specifics of prokaryotic translation, there are a couple concepts that are very important to understand. The first concept that you should understand is the tRNA and the amino acids and their interactions with one another. And we talked about tRNA structure. We also talked about how the specific amino acid finds or binds to the tRNA, to its specific tRNA. And then we also talked about how these tRNAs have the ability to bind to different codons because of the last codon, which is the wobble-base hypothesis. We also talked about amino acid activation. Amino acid activation is a process when we have the amino acid just loaded onto its tRNA, and this process requires two ATP equivalents, which are two high energy phosphate bonds. And then we talked about something that personally fascinated myself personally a lot, which was the amino acid tRNA synthase enzyme. The synthase enzyme is specific to every amino acid, and we talked about how or why different amino acids can find their specific tRNAs, even though the nucleotide sequences are the same. The CCA nucleotide sequence, which we talked about at the 3' end, are similar for all tRNAs. So we had a question: how do these amino acids just find their right tRNA? We talked about that in this video right over here. And we also talked about ribosomes. And we talked about ribosomes, the difference between prokaryotic ribosomes and eukaryotic ribosomes. So please make sure you watch these videos and understand all these videos before you have the chance to watch this video, because this video will need the necessaries of that video for you to understand everything. So without further ado, let's get into it. Prokaryotic translation is really something very different from eukaryotic translation, and that's because the internals, they have no internal organelles. They have nothing. They have no organelles inside the cytoplasm. Therefore, everything has a chance to interact with one another. So the question I'm trying to ask is, what is holding transcription and translation from happening at the same time? The answer to this question is absolutely nothing. Transcription and translation can happen at the exactly same time simultaneously. So this is what it looks like essentially. So we have the DNA right over here, and we have an RNA polymerase, which is going to be synthesizing or which will be transcribing the mRNA. The mRNA has three different sections. We have the first part, which is the initiation sequence. We have the open reading frame, which is the main body of the mRNA. We have the termination sequence, which just highlights that the mRNA is read, and the ribosomes can then dissociate. So the question I want to ask is I have it over here, but identify the 5' and the 3' ends. I want you to think about this. If you were trying to identify the 5' and 3' ends inside an exam, the first question you would ask yourself is, what direction does translation occur in? Translation happens in the 5' to the 3' end. And because we can see that we have initiation sequences over here, and the ribosomes are moving in this direction, we know that this is the 5' end and this is the 3' end. And it makes sense that because the synthesized mRNA has not produced the termination sequences, that's why we can have these ribosomes being, just having polypeptides being formed and peptides being polypeptides being formed. So this is very essential in understanding the mRNA and translation. This is a very simple question that I've seen on IB exams, and I'm sure this is going to be a major understanding component when it comes to the MCAT exams, in terms of the content. So, does translation always begin at the 5' end? And if not, what does it suggest? So this question is asking, is it always necessary that transcription or translation has to start at the 5' end? I'm not talking about the initiation sequence. I'm talking about the 5' end. What this means is that, does this ribosome really have, if it's coding for a specific enzyme, does it need to start from the 5' end over here? The answer to that question really lies in the understanding of what polycistronic is. Polycistronic enzymes are. Polycistronic enzymes polycistronic mRNAs are really mRNAs which have more than one gene on it. And if you look at the word poly, it means more, and cistron means gene, so more than one gene. And how does this exactly look like when it comes to translation? And it looks something like this. So we have two different open reading frames, and they will code for their own polypeptides, and you can see that by the different colors over here. So it's not true that we don't need a gene to be synthesized, or a polypeptide to be synthesized from the 5' end. You can have it being synthesized from the middle as well, or a different part of the mRNA. And sometimes we have a higher promoter. We have a lot of promoter sequences sometimes, and there are variations. So the answer to that question is no. It doesn't always need to start at the 5' end, and that's because we have different genes inside prokaryotes. Prokaryotes often have polycistronic mRNAs. So you can imagine that. Let's say we copy paste this into a larger mRNA. You probably have a lot of different genes on it, so it'd be producing a lot of different polypeptides, and that's why that's a feature of a prokaryote. And eukaryotes almost always have monocistronic. For the sake of the MCAT, we are told to memorize that the eukaryotes always have monocistronic mRNAs, and that's because they're more sophisticated and they have to go through splicing and whatnot. So they have a more sophisticated response in comparison to prokaryotes. So now let's get into more detail of the mRNA, and let's focus on the different parts of the mRNA in terms of the sequences on it. So the first sequence in the polycistronic, does translation begin? Well, we talked about this right now. Let's not talk about it. The question is exactly asking, does the polycistronic translation occur exactly on the 5' end? And this just means that the first nucleotide on the polycistronic mRNA is this where translation really starts? And the answer to that is no. So we have to have the formation. So there's a couple sequences on the mRNA which will tell the entire enzymes and everything, or the ribosomal proteins, that this is where we want to start translation. And those sequences inside prokaryotes are called Shin-Dalgarno sequences. And this is just for prokaryotes. Do not confuse this for eukaryotes. They have something else. The Shin-Dalgarno sequence is located at -10, and -10 is just referring to upstream 10. And we talked about the difference between, we talked about upstream and downstream before. So the Shin-Dalgarno sequence is located at the -10 over here. So the Shin-Dalgarno sequence, its complementary base pairs are found on the small ribosome. Are found on the ribosome. So this allows the attachment of the ribosome to the mRNA, and this will initiate the entire sequence or the process of translation. So just like transcription, translation has 3 steps. It has initiation, elongation, and termination. And I'm sure that you're aware of the overall concepts of what's happening in initiation, elongation, and termination. Initiation is just when translation is started, and elongation is when the polypeptide is growing, and termination is when the polypeptide has stopped growing and the mRNA is read. So what is exactly happening on a molecular scale in initiation, elongation, and termination in prokaryotic translation? And that's exactly what we're going to talk about now. So initiation, we first have the small ribosomal subunit. We're going to have IF factors 1, IF factors 3 proteins that will bind to the small ribosomal subunit. And with this, we're going to have the mRNA attach on it as well. The second thing that happens is we have the IF2 factor that joins with the first amino acyl tRNA. So the first tRNA is going to bind to the P site, and with that we're going to have the IF2 factor join with it. This step requires energy, and there's a source of energy, of course, like everything has a source of energy, and we're going to talk about that in a second. The last thing that happens is we have the 50th large subunit that will come and bind on to this entire complex. So now we have the initiation complex completed. So here's the question. every unfavorable reaction requires energy. So this is an unfavorable reaction. According to thermodynamics, this is not going to happen naturally. There is energy that's required for this reaction. So where is that energy coming from? This energy comes with the IF2 factor. The IF2 factor contains a GDP molecule. This GDP molecule will be hydrolyzed to GDP, and this allows the attachment of the first tRNA to the P site. But you might be thrown off by what a GDP is, and that's totally fine. The only difference between an ATP and GDP essentially is the aromatic ring structure. Or the aromatic nitrogen base. And what I mean by that is if we were to replace this adenine with a guanine over here, so this is adenosine triphosphate, the adenosine and the triphosphate, and if we replaced this with a guanine base, this would simply allow us to have a GDP. So the hydrolysis of a GDP will likewise create energy. And this really just, if we understand what this means, is phosphate hydrolysis releases energy. So when GDP is hydrolyzed, we have energy being released. So this is where our energy is coming from to bind this tRNA to the P site. Another thing we want to talk about is the first tRNA. The first tRNA is a special tRNA. It's an initiator tRNA. And what makes the initiator tRNA different from other tRNAs? The answer to this question lies in its amino acid. The first tRNA is the initiator tRNA, contains an amino acid with a modified methionine base called the formal methionine, the FMET. The FMET is a very special amino acid in the sense that it indicates that this is if you look at a number of different prokaryotic polypeptides, the first amino acid that is found to be the FMET. So the FMET is very important in that sense. It helps us recognize that this is a prokaryotic polypeptide, and this is where the polypeptide chain started. So this is something you should memorize at this point. And we're not going to talk about the chemical aspects of the formal methionine, but this will be discussed in biochemistry and the chemistry section. And hopefully I have that on my channel by now. So the next thing we want to talk about is the start codon. The start codon is the AUG. And now our mRNA is going to have the AUG on it. The AUG is essentially indicating that this is where transcription or translation has to start. So we have talked about the Dalgarno sequence a while ago, and a question to ask is if translation occur if the Dalgarno sequence is not recognized by RNA? And the answer is no. RNA will not be attached to the mRNA. You cannot recognize it. So everything has to be recognized by the mechanisms in the initiation complex for proper initiation to be started. And if recognition of the Dalgarno sequence doesn't happen, then we have translation failed. So this is when we have the initiation complex completed. And before we proceed into elongation, all these proteins, these initiation factors, will fall off. And this is when we have elongation starts. So elongation will begin, and there's a 3-step cycle to elongation. The first step is when we have the tRNA. We're going to have the first tRNA come to the A site. It's going to form a hydrogen bond as well over here. And this also requires energy, just like we needed energy with the first step, first tRNA binding with the P site. And this energy comes likewise from GDP. And the hydrolysis of GDP happens by something called the EFTU factor. The EFTU factor is an elongation factor which will hydrolyze GDP to GDP. So what I mean by is guanine triphosphate to guanine diphosphate. And once this happens, we have the GDP sitting on the EFTU factor, and this has to be resetted, because if this EFTU factor, elongation factor T2, has to hydrolyze another GTP, then it needs to be resetted, and that's resetted by the EFT, which allows the removal of the GDP. And I'm trying to say GTP here, not GDP. I just want to fix that. It's annoying my soul. 1 second. GTP. Okay. The second thing that happens is peptidyl transferase. Peptidyl transferase is really just the process through which the peptide will start to grow and the tRNA will go into the next position. So what happens essentially is the F-met amino acid will nucleophilically attack the other peptides. So if we think about this, all that's happening is in a sense that the peptidyl transferase activity. So what I'm trying to say essentially is that the second tRNA will nucleophilically attack the tRNA of the other nucleophile. So what's essentially happening is there's a nucleophilic attack. So if you want to understand the exact mechanism behind this, I recommend you pause this video and you go and you look up the video, how peptide bonds on my channel, and you'll understand the chemical aspect of how this is happening. So we see that there is a carboxyl group over here, and there is a amino group here. The amino group is going to be nucleophilically attack is going to nucleophilically attack the first amino acid, the F-met, and this is going to form a bond, or the peptide bond between these two, and this is also mediated and catalyzed by the 23S RNA, the ribosomal RNA inside the large ribosome, and this will allow the entire formation of the peptide. So we know that, okay, so this is a good way to memorize. So we have a nitrogen terminal, we have a amino terminal, and we have an amino terminal, and we have a carboxyl terminal inside amino acids. And if we understand how peptide bonds are formed, we will know that the first amino acid will have the amino group terminal in the start, and then we will end with a carboxyl group. And this is the directionality of the growing polypeptide. The last step is called translocation. Translocation is when we have the movement of the tRNA. So what's going to happen is the first tRNA is going to move to the second site, and the second tRNA is going to move to the E site. And you might be asking, why do we need the first tRNA to be on the E site? And that's because it's thought that the E site, or the tRNA without the amino acid on the E site, will stabilize the entire mRNA inside the ribosome. And it's been found that if there are any mistakes in this process, it encourages frame shift mutations, and this can cause a major impact on the amino acids that are being synthesized. So therefore, this is very essential in a way. So translocation happens with the help of the elongation factor G. And this is also called a translocase, ASE at the end, which just shows it's an enzyme. Now this process can repeat again and again. So we just repeats it again and again. We have another tRNA that will come over here. And this process is called elongation. And this process will essentially keep on happening until there is a termination sequence found. And the termination sequences are recognized by the RF1 and the RF2, which are proteins called which are enzymes, which are called release factors, and they help recognize the stop codon. So the release factor 1 will recognize the UUA and the UAG patterns, and factor 2 will recognize the UAA and the UGA. This is the end codon. This is the end codon, and this is the UUA. So UUA is going to be recognized by the RF1 factor, and this is going to allow the entire ribosome to fall off. How much energy is required to synthesize 100 amino acid polypeptide chain, including amino acid activation? If you've seen the amino acid activation video, you know that the amino acid activation requires 2 ATP equivalences. An ATP equivalence is just a high energy phosphate bond. And we're saying that now because if you remember, elongation and initiation elongation requires GDPs as well, so it's just reference to a high energy phosphate bond. So amino acid activation requires 2 high energy phosphate bonds, and we have elongation. The first, we have the tRNA, the new tRNA moving into the A site. That's 1 ATP equivalence used there, 1 high energy bond is needed. So the GTP, the GDP is hydrolyzed to a GDP, and that's how we have one phosphate, high energy phosphate used, and then we have a second one that's used during translocation of the tRNA. So that's two per cycle, And then we have initiation when we have the first tRNA that binds to the subunit that requires one energy as well. So that's one. And then we have a termination sequence, and when the termination sequence is when the last codon is read, there's energy that's required to that is required to expel the last tRNA. That also requires one high energy phosphate bond. So altogether, if we look at this, so let's think about it now. We have all the information we need. So we have 100 amino acids. So if we have 100 amino acids, then if there are 2 phosphate bonds for every amino acid, that's 200 right over there. And then elongation, we have 99 elongation steps. That's going to be 100, so 99, let's do the math over here. So we have 200 from elongation, from amino acid activation. And we have 99 times 2 is, let's see, 198 actually. Yep, there you go. Can't even do math. 198. And then we have initiation, which requires 1, or the initiator tRNA needs 1, and then the termination sequence needs 1 more. So that boils down to 400. So this just means that this is just 4x, and we have now understood why the number of amino acids will require 4 times the amount of high energy phosphate bonds. And you can memorize this, but hopefully understand why it's 4x.